differential gene expression influences function and small RNA regulates gene expression. So, when we think about our own cells, we have the same genes in every single cell of our body. However, we have different gene expression. So, although every single cell might contain an albumin gene and a crystalline gene, not every single cell expresses those genes. And that's what leads us to cell differentiation. So we can have epigenetic changes which turn off certain genes, but ultimately we can also have different gene expression because of available activators. So a liver cell is going to be very different than a lens cell. So a liver cell will express albumin because it has the activators necessary to begin this transcription. A lens cell will transcribe crystalline but not albumin. So liver cells are not going to make proteins that are important for refraction and transparency, and a lens cell in your eye is not going to make blood proteins. So it comes down to which activators are available, and that leads to different tissue-specific proteins being created, and we have therefore different gene expression and cell differentiation. So again, we have these different genes. We have the same genes, but different proteins are made. And if different proteins are made, we're going to have different structure. And if we have different structure, we're going to have different function. And if we think about this, our liver is made up of hepatocytes, these little liver cells. And our liver does make bile, which helps with the emulsification of fats in our digestive system. But a big part of our liver is to process nutrients as they get absorbed through our intestines. They head straight to our liver for processing prior to being headed back to our heart and pumped to the rest of our body. Another thing the liver does is that it makes all of our blood proteins, and albumin is one of those. So if we think about our blood, we have proteins that are floating through our blood besides red blood cells. However, we also have a lot of water in our blood. And again, our blood cells, our capillaries, are touching every other cell in our body. So in order to keep the fluid nature of our blood, we have to keep water in our blood. Albumin is too big to go through the capillary walls, and it also attracts sodium ions, which then keeps the solute concentration. If we think back to osmosis, it keeps our solute concentration in our blood high so that the water stays here and it doesn't rush towards higher solute concentration in the cells that are bordering the blood vessels. So ultimately, albumin does help us keep this osmotic pressure and keeps the water in our blood, but it also, in addition to attracting and carrying ions, it will carry drugs in our body, and it will also carry hormones and transport many different things through our blood. So again, because our liver cells are making albumin and blood proteins, it has a very specific job, and it comes back to the structure of those cells. Now, if we think about our eye, we have these lens cells, and this lens cells is what takes in the light and refracts it to our retina so that our brain and our optic nerve can make sense out of what we're seeing. And again, if we think about our lens cells similar to our skin cells, we have this basal layer of mitotically dividing like germ cells. So these cells are active and dividing, but as it goes deeper, unlike our skin cells, which make a tremendous amount of keratin and tough keratin protein for our tough outer skin, these cells are making a tremendous amount of crystalline. So the crystalline builds up in these cells, and these cells actually lose their nucleus and their organelles, and they're just packed full of this crystalline, and that crystalline, it allows it to be transparent and also refract light. Now if we think about this, these cells in our eyeball are not going to make blood proteins, just like our liver isn't going to make crystalline protein. So we have the same genes in both of these cells, but they're going to make very different proteins, and the cells are going to have different structure, and they're going to carry out different functions. So although they have the same genes, through differential gene expression, they're going to end up with different structures and functions. Now, as we talk about all this expression and regulation, we spend a lot of time talking about turning on the gene at the transcriptional level, talking about our promoters and our repressors and everything like that. However, as we go in, and there are many other regulation levels to this, and a lot of them involve small RNA. So when we have alternative splicing, we have these small nuclear RNA molecules that end up becoming part of our spliceosome, and our spliceosome is what ends up cutting out the introns and can alternatively splice our exons in order to express different proteins from the same transcript. So again, we can have A, B, C, D, E as exons within the pre-mRNA, these transcripts, but then we're going to splice out the introns, and we can reconnect the exons in different ways. In this protein, it's made from the exons A, B, C, D, and E, whereas this protein is going to have a different structure and a different function because it's made of A, B, D, and E. So with C being spliced out along with the introns, you can have the same gene, but then ultimately end up with different proteins, which will have different structures and different functions. Now, we're always talking about turning genes on or preventing them from being transcribed. Now, part of it is like after we have the genes turned on, how do we turn them off? And again, every time the DNA is read, a lot of times we're making thousands of messenger RNA transcripts, and then all those thousands of messenger RNA transcripts are going to thousands of ribosomes and making countless amounts of protein. Well, that protein is carrying out function, and sometimes we want that function to stop. So we need to be able to turn off the RNA, and we need to be able to turn off the proteins. And small ribosomes or small RNA has a role in there. So usually we think of RNA as single-stranded, but we can make this double-stranded RNA, which a dicer enzyme can come and split it up and cleave them into smaller pieces. So now we have this double-stranded RNA, but now it's cleaved into these small RNAs. And these small RNAs can form different things. So we have this silencing complex, and this small RNA can get involved with these proteins and then interact with specific messenger RNA transcripts and break down the transcript, so then it's no longer being translated. These other microRNAs can hook onto the messenger RNA, and it doesn't necessarily degrade it, but by hooking onto the messenger RNA, it can't feed into the ribosome and be hooked up and begin translation because it kind of blocks it from getting into the ribosome and actually being translated. So again, let's say we get to that point where we've created all these proteins. Now again, if we no longer want that protein to be functional, we can degrade it. So the protein can be tagged, and that once it's tagged, it gets sent to this proteasome, which is a multi-protein complex. And that proteasome can then break down the big protein into these small polypeptide fragments that are no longer functional and don't have that three-dimensional shape that makes them work. So again, differential gene expression is going to influence the function of a cell, and small RNA play a major role in regulating gene expression.